Hello everyone, Dr. Paul Rose again, and in this presentation we're going to be looking at how animals in zoos can have their husbandry based on important features from their wild ecology and how we would go about looking at features of their wild ecology in what's provided in their enclosure. This practical focuses on ungulates, a diverse group of mammals that are naturally found in all of the world's continents apart from Australia and Antarctica. Ungulates are commonly seen in zoos. They occur in two basic types, the ruminants or even-toed ungulates, which chew their cud, and the perissodactyla or odd-toed ungulates, which ferment their food in the hindgut. So a different strategy to the artiodactyla or ruminants. We could also talk about the sub-ungulates as well, such as the elephant, as some of their enclosure features they're commonly provided with are similar. Ungulates are normally sociable, they have a particular social system, and often, as in the case of these fallow deer, they are sexually dimorphic. The males have ornamentation for sexual display and competition. Many species of ungulate occur in specific niches in the wild. Here is a Sitatunga, which has been provided with access to a wetland that only that species can access within this mixed taxa exhibit. Sitatunga are a wetland antelope. They are adapted with very long hooves for marshy environments. They like cover and thick vegetation, and therefore this is an example of an important biologically relevant resource in the enclosure. Looking at biologically relevant features of an animal's enclosure is obviously something that we don't just do for ungulates. We should do this for all species that we look after. What causes a particular resource to be highly valued or highly used by the inhabitants of a zoo enclosure? These sacred ibis wandering around in their pool, for example, they are a wading bird. This is the environment that they would forage in. So knowledge of the species' wild ecology and its behavioural ecology can help us provide the right resources in captivity. And sometimes highly valued resources in the zoo might spark competition or aggression. If we feed a species in a different way to how it would feed in the wild, concentrating flamingo pellet in bowls, for example, we could see behaviour change that differs from the normal activity pattern we would see in a free-living animal. And when it comes to carnivores, or species that range across a wide area of their territory or home range when they look for food, we might have to provide a complex array of enclosure furnishings and resources to reduce any anticipatory behaviours, to reduce any abnormal repetitive behaviours, and to promote natural feeding and foraging strategies to enable the outlet for these highly motivated behaviours that we might see in the wild. These are wolverines, a species that can have a large home range of many hundreds of square kilometres in the wild. The wolverine is an active carnivore and a scavenger. It will travel around its territory regularly in search of food, marking its territory too to keep away rivals. So a diversity within its habitat and its husbandry regime is important in the zoo to ensure it doesn't promote abnormal repetitive behaviours. The ungulates are a useful set of species to study from a positive welfare aspect because they do lots of behaviours that tell us they are contented in their enclosure. Chewing the cud, for example, as in the case of the Sitatunga. Resting or lying down in a social group indicates that they're also not being alert or vigilant all of the time. So we can assess the resources that are provided alongside of the wild behavioural ecology of the animal. What types of habitat should it occur in the wild? And is this habitat replicated in the zoo? Are the animals spending normal amounts of time feeding or foraging, as we would see in the wild? Or are they spending excessive amounts of time being alert or vigilant because they might be unsure of their surroundings. This practical is based on a paper by Philip Jarman, and this paper explains the social organisation of antelope in Africa in relation to their ecology. And their ecology is based on their social grouping, on their habitat preferences, and on how they collect their food. You can have a read over this paper if you search for the title and author in Google Scholar. The paper as a PDF file is free to view. And Philip Jarman breaks down the various species of African ungulates into those that are browsers and grazers or intermediate feeders, into those that occur in open grassland, those that occur in bushland, and those that occur in dense undergrowth. So he's comparing the foraging strategies of the animal with the habitat that it's found in. And alongside of this, he looks at the different social systems that the animal occurs in. Those that live in the open plains are going to be found in much larger groups than those that live in dense undergrowth, where there is less space for a large herd to form. So we want to test this idea in the zoo. We're going to look at different species of ungulates. We're going to look at their social organisation. We're going to look at the habitat preferences that they exist in the wild. We're going to look at their feeding and foraging strategy. How do they collect their food? And we're going to judge the quality of their enclosure that they have been provided with based on this ecological information. And we're going to measure behaviour, alert or vigilant, that might tell us about welfare. I'm now going to explain the instructions for the practical. I have filmed the behaviour of 6 species of ungulate, all housed in the same zoo. All of these species naturally come from Africa, and we're going to be looking at differences in their wild ecology, the habitats that they come from, the social systems that they would live in, and their foraging behaviour as predictors of what they might do in the zoo. We're also going to look at the space and type of enclosure that they're provided with too. So the first thing you need to do is look up each species and note down key information on the habitat preferences, i.e. the habitat they have evolved to live in the wild, their foraging ecology, i.e. how do they collect their food, and their wild social grouping. Are they solitary? Do they live in pairs? Do they live in large herds? So on and so forth. The IUCN Red List, simply Google IUCN Red List and the page will appear, is probably your best source of information for this background ecological facts. The 6 species of animal are the Grevy zebra, the Sitatunga, the white rhino, the common waterbuck, the scimitar-horned oryx, and the gemsbok. So 6 species of African ungulate, all with different ecological backgrounds. What do we need to know about them to keep them properly in the zoo? You'll need to watch each video in turn, pausing so you can code the animal's ecology and habitat based on what you see, and then you will count 2 different behaviours as they occur. So for each video, record the species that you are watching. Give the species' natural habitat a code. Your knowledge of its natural habitat will come from the review that you've done in point 1. O would mean open grassland, B would mean bush, i.e. the kind of thicket that we see within the grassland itself, sort of open woodland, and D would be dense cover, i.e. species that might occur in thicker woodland or forest. Give the species' foraging ecology a code as well, and this is very well studied. So G would be grazer, I would be intermediate feeder, and B would be browser. Grazers only eat grass, browsers only eat leaves from trees and shrubs, intermediate feeders do both. Give the species' social structure a code. Again, you will find this information out from your review of key ecological facts from point 1. Is it a solitary species? Does it live by itself? Is it generally found in pairs or small groups? Or does it live in herds or larger groups? So what type of social system should this animal live in? And finally, your last code to do is the species enclosure in the zoo. Give it code A for simply flat pasture land. Give it code B if it's some pasture land that has some cover, maybe some groups of trees, for example, or bushes. Give it code C if it's well planted and furnished with a range of objects that provide cover in a different number of areas. Once you've done all of that, start watching the video and count the frequency of these two behaviours. The number of times you see vigilance or the animal being alert, i.e. the animal's head is up and it's looking around. The number of defined feeding bouts is the next thing to count. Head up, head down, consuming food. So when the animal is feeding, that would be one bout. If it changes behaviour and it does something different to alert, you wouldn't count that. But if it was feeding and then it suddenly looked around itself, you would count that as an alert behaviour. So we're not timing here, we're just doing a simple tally of the number of times we see a feeding bout and the number of times we see a vigilance event. Point 1 provides you with your wild evidence to evaluate the zoo. Points 2, 3, 4, 5 and 6 give you an idea of how you're going to measure this evidence to evaluate what you see in captivity. So you've got your reliable source, which is the IUCN. You've coded how that information might be assessed in the zoo. You have then assessed what the animal gets by coding the provision within the enclosure. And then you have data that give you an idea as to whether or not the zoo is providing an ecologically relevant exhibit. So here we have a methodology for taking evidence, for coding that evidence, for asking the animal, and for evaluating how provision is used for the animal in captivity. This is what your raw data sheet should look like. Copy this format in Excel for your animals. These are fictitious data, and I haven't related any of my codings to any particular species. So don't copy any of my values or letters here. Simply use the same format with the codes in the instructions. Individual is a continuous list of the individuals that you study. You will need to record behavioural data for each individual animal that you see in the video. Again, don't worry about timing, just count the alert events or feeding bouts for each individual. So there are 6 species, as I said, so I've made up that there are 20 individuals. I've got 4 individuals for species A, and I've got 3 individuals for species B. I then coded the habitat that they would live in the wild, O for open, B for bush, C for cover. I then coded their foraging strategies of grazing, which is the G, intermediate feeder, which is the I, and browser, which is the B. I have then looked at their social structure, H for herd, P for pair, and then S for solitary. And there's my codes for the enclosure. The pasture land is A, the pasture land with some features is B, and the pasture land with varied cover is C. So do that in exactly the same way, but actually code that information correctly for the species that you're looking at. And here's my fictitious data of counts of alert events and counts of foraging bouts. So this is how you would put your data into Excel before lifting those data into Minitab for analysis. So we're going to do some different types of analyses on these data. So for descriptive statistics, we're going to look at whether or not the enclosure style matches the habitat requirements for the species. We're going to look at mean times spent, or mean frequencies, I should say, on those behaviours compared to the habitat provided and the enclosure style they get. We're going to look to see if foraging ecology is catered for in the enclosure design, if we've got differences in time spent foraging, is that predicted by the style of enclosure based on the species' wild ecology? We could also look to see if their social structure is similar to the wild evidence we have gathered. So you would simply count the number and sexes of the animals in the group if sex is identifiable. For example, this is a female Sitatunga. The male will have horns. And we would also look at what percentage of species from this overall study are housed in a biologically relevant manner. So you could work that out from the end of your analysis. How many significant p-values do you have that suggest that what we know from the wild is predicted based on the evidence we have gathered, and is that replicated in the zoo? So what we're going to look at in Minitab is a Poisson regression, because this deals with count data. And we're going to see if foraging and vigilance, these two behaviours that you've counted as a frequency, are predicted by how the enclosure has been scored, as well as key aspects of species ecology, for example, their wild habitat. We're also going to see if foraging and alert or vigilance correlates. So we're going to run a correlation test, and we're going to test our data for normality to make sure that we know that our correlation is appropriate for the spread of data that we've got. We're also going to look at some descriptive statistics, and we're going to look at some post hoc testing as well, so that if we do have any significant outputs from our model, from our Poisson regression, we can see where those significant differences lie. Please remember that this is what your raw data sheet should look like, having watched the videos of the 6 species. You should have coded the habitat type, the foraging strategy, the social grouping, and the enclosure style for each individual of each species. And you should have individual behavioural scores of alert and of foraging of each individual that was viewable in the video for that species. So open Minitab and paste in your data. Remember that these codes are what are going to tell us about the different characteristics of the species, their ecology, and the enclosure, but my alert and foraging frequencies are fictitious data. I've made these up for the purposes of this practical. The first thing we need to do is to see whether or not our behavioural data are normally distributed. So we go to Stats, Basic Statistics, and we go down to Normality Test. We place in each behaviour in turn, and we look to see whether or not the resulting p-value is greater than the 5% level. If it's greater than 5%, that means the data are normally distributed. So my p-value of 0.944 is greater than 0.05, it's greater than the 5% level. So these data are normally distributed. Let's do the same for foraging. And that too is greater than the 5% level, so is normally distributed. So we want to see whether or not our behavioural terms correlate with each other. Does being alert influence the foraging activity and vice versa? This is important because it means we can select for specific terms later on in our modelling, knowing whether or not there is any relationship between them. So we're going to go to Stats, Basic Statistics, and Correlation. Because we checked our data for a normal distribution, we can use the Poisson's correlation. If our data were non-parametric, were not normally distributed, we would use the Spearman's correlation. So let's place in our 2 behavioural variables and click OK. And here you can see there is no correlation between alert and foraging. That means there's not likely to be any collinearity between these variables, and that could push up a variance within the model, what's called variance inflation, and make our model less reliable. To show what that means by looking at the data visually, if we just draw a simple scatter plot and we pop a regression line on it, and we add in our 2 variables that we think might be related, we can see that there is really no slope to that line and really no patterning to how alert and foraging interact with each other. So we can assume that these are 2 independent behaviours. So what we're going to do now is think about how can we determine differences between the individuals, the species of animal, the various ecological measurements that we've taken, and the enclosure provided at the zoo. How might that impact on behaviour? It's always useful to get some descriptive statistics of your different categories so you can see why things might be patterning or distributed in the way that they are. So I'm going to go to Stats and I'm going to go to Basic Statistics, and I'm going to display descriptive statistics. And again, I'm interested in alert, and I'm interested in the habitat of the animal in the wild. I'm going to go to this extra button called Statistics, and I'm just going to ask Minitab to just give me the mean and the standard error of the mean. If your data are non-parametric, if they weren't normally distributed, you should use a median as your best measure of an average for your data. And then you would also use a standard deviation. So here we can quite clearly see that there are differences in the mean frequency of being alert based on the open, the covered, or the bush-like habitat. What about foraging? Do we see a similar pattern when we look at habitat type and time spent foraging? There doesn't seem to be any real difference. 4.7 would round up to 5, which is very close to 6, and 6 is very close to 8. And the standard error is all very similar. So clearly foraging isn't particularly related to habitat type, but when you are looking around being vigilant, there might be some kind of relationship. So how does that affect enclosure style? So let's go back to alert, and let's say, well, does this show a similar pattern with enclosure? And here we can see quite clear differences in the amount of cover provided to the animal and how alert they might be. So it's important to consider what's provided in the zoo, why we might see differences in the values that we have, and how might that be explained by wild ecology. And to get a better explanation of wild ecology, let's do a model that might suggest why some of these differences are here in the first place. We have count data, and one of the best things to use for count data is a Poisson regression. And Poisson regression can be found under the regression tab here. So we go to fit Poisson model, and we say our response variable is alert, and our continuous predictor is going to be the individual animal, because we're interested in individual differences. And our categorical variable to start with is, let's say, the wild habitat type. So are there differences in alert behaviours that could be predicted by individual differences or by habitat quality, habitat preference in these animals? We click OK, and we look at our model output. We can see that the p-value for the individuals is above the 0.05 cutoff, so individuals don't differ in their behaviours. So that's clearly within those categories, we've got no real individual difference. But for habitats, that does seem to be predictive. So the individuals within the different habitat types, whilst the habitat is predictive of the behaviour change, the individuals within each category of habitats seem to be behaving very relatively similar. We look at our R-squared value, which gives us an idea of how reliable our model is. And this R-squared value, and we always use the adjusted value, is 41%. So this suggests that our model is quite reliable. And then we have some constants, so coefficients of the model, which give us an idea of where the model is likely to go in the future if we added X values to being alert based on that category. So we look at the coefficients for a covered habitat, and we can see that alert is likely to increase or decrease based on the coefficient. So being alert suggests that you are going to see a decline in being alert by minus 0.03 in an open habitat. So you're less likely to be alert. And we look at our variance inflation factors as well. And ideally, we want them to be around 2 to 3. Around 1 is best, but this is behavioural data, and behavioural data is always messy. So that's a model that's looked at individual differences, and it's looked at the habitat of the animal. Let's run another Poisson regression. And let's this time change habitat to something different. Let's now consider whether or not the foraging strategy of the animal in the wild, is it a grazer, is it a browser, is it an intermediate feeder? Does that affect the time or the frequency of alert behaviours? Perhaps if you're a grazer and you spend more time with your head down, you're more likely to have to move your head up to look around the environment. And again, individuals themselves don't differ within those categories, but foraging does seem to predict being vigilant. And what we can see here is actually an increase in being alert if you're an intermediate feeder compared to a grazer. Now, some of you might realise that we've only got coefficients for not all of the categories of terms that we've included. And that's because it's taking one of the terms and comparing the others to it. So in this case, being a browser, the differences from being a browser to an intermediate feeder and a grazer are being compared back to a browser. But if you're interested to know all of the differences and where they might lie, you can look at that by going back to your model, by going to your factorial plots, and by asking it to draw you a plot of the particular variable which you've put in, so that's the individual animals and the foraging activity, against the response, which is alert. And if we click OK, we can see that we've got the mean frequency of being alert, browser, grazer, and intermediate feeder along the bottom. And clearly, the intermediate feeder is much higher in the time spent being alert compared to the browser or the grazer. So we've got a significant difference between the intermediate feeder compared to the browser and the grazer, which probably don't differ. And I'm going to do one last Poisson regression. And this time we're going to look at a zoo factor. So let's now say that we've looked at categories that relate to wild ecology, so the feeding strategy or the habitats that the animal has evolved to live in. Now let's think about the time spent foraging and whether or not the enclosure is providing for these species-specific differences in foraging strategy. So I'm going to put enclosure as my categorical predictor, and I'm going to click OK. Again, individuals don't differ, but the enclosure themselves is also non-significant. We've got a good R² value of coming up to 40%. And again, we can see that our coefficients, whilst they might look different, are not actually significant. So let's go and draw another graph of that so we can see what's going on. So our response was foraging, and our enclosure is the one that we're interested in. And we click OK. And what we can see is whilst the differences in enclosure might seem to suggest that those animals in more covered enclosure have a higher foraging time, overall, there's no real significant difference between 8.9 maybe, 4.5 and 6. So the numbers themselves don't suggest any significance. Now we've got some normally distributed data here. So what we could do if we wanted to see a little bit more detail into why we might not have an effect is we could go to ANOVA and we could run a one-way ANOVA, and we could say, let's look at foraging against enclosure time, and let's look at some comparisons and ask it to run a 2 keys comparison and see what happens. And then we have this really nice graph that gives you a bit of a clearer explanation as to what's going on. And you can see the overlap between the error bars on the graph. So this graph here is explaining the Poisson regression. It's explaining why those numbers might look different, but actually the range around the numbers overlaps, the error bars overlaps. So the chances of the data point for A, the data point for B, and the data point for C differing are actually relatively slim. So just to repeat myself with what I've just said there, let's go to the project manager output so you can see what we did. and I asked it for a main effects plot, which I would like it to open, and I would like it to open my interval plot. And let's pop those next to each other. So the Poisson regression is really useful for using for count data. So if you ever have counts, I recommend a Poisson regression. But to better understand the patterning within those values, those factors that you're inputting, if you do a two-keys post hoc analysis and you look at the interval plot, it can help you explain why differences, so A and B, are looking different to C, but they're not significant. This is a lot clearer to see because the overlap of the error bars suggests that the variation around the mean is going to make each particular value for your different styles of enclosure not significant. So what we've done here is we've looked at a correlation and we've looked to see whether or not we can predict the future, what's likely to be the increase or decrease in alert or foraging behaviours based on the categories of a variable of predictor that we've included. And we've looked to see why there might be differences in the amounts of time spent, or the frequencies, I should say, of these behaviours based on ecologically relevant factors, i.e. the natural habitat of the animal, compared to what we see in the zoo. So in this case, with these fictitious data, natural ecology suggests that animals should do things differently, but in the zoo, the enclosures seem to be relatively standard and are not providing for those particular outputs. So we could suggest changes to the zoo's enclosures that might help them become more naturalistic for the animals that we are displaying. Again, I've made these data up. Your data might look totally different, but I suggest that you input your data into Minitab, you run a normality test, you do your correlation, you look to see what descriptive statistics show you of the means or medians, depending on the distribution of your data, are for your particular ecological categories and for your enclosure category. And then you run a Poisson regression on your count data to see what might be predicting why these differences are the way they are. So why is it that we have to consider ecological requirements in the husbandry of the species we keep in the zoo? We could revisit our original sources, the IUCN Red List website, as well as Philip Jarman's paper on social organisation, foraging strategy, and where you find these species in the wild. What is their habitat preference? All of those wild information can be compared against the behaviours that you have seen across these different species of ungulates that are all housed in the same animal collection. We can then get an unbiased and objective judgment on enclosure quality and on enclosure design that impacts on important behaviours. Ungulates spend a large part of their daily time activity budget foraging. We need to promote foraging activity in the zoo. This is essential for the health of their gastrointestinal tracts and the microorganisms that allow them to digest their food. Rumination, for ruminant herbivores, for the artiodactyla, also keeps them calm and relaxed. So we must always promote rumination too. High levels of alert behaviour might be indicative of animals that are not contented in their environment. This might be impacting on the time spent foraging or on the time spent ruminating, and therefore we could look at the ecology of the animal, what does it need from its environment, and alter the enclosure accordingly to try and promote these welfare positive behaviours. I hope you've enjoyed this practical and the explanation of using ecological knowledge to understand husbandry and provision and to provide a more biologically relevant environment for the animals that we keep in the zoo. Thank you.